Okay, welcome back, everyone, to the FARA Flash Talks. I'm Liz Sorani. As we continue to celebrate FA awareness during the month of May, today we're hosting 5 new Flash Talks presentations from our junior investigators. They have 5 minutes and 1 slide to tell us about their research. And after each talk, we'll have 5 minutes of question and answer. And you can submit your questions or comments by using the Q&A box at the bottom of your screen and typing into the box. All the sessions, oops, I should have changed my slide. There it is. Are recorded and will be posted on FARA's YouTube channel and are also posted on the news page of FARA's website. After the webinar, you will be prompted to follow a link to a survey. You'll be asked to rate today's presentation according to a few criteria, and we'll put also the link to the survey in the Q&A box or the chat box. At the end of the series next week, we will recognize the best presentations with an award. Before we get started, I would like to ask our co-moderator today, Joe Tyler, to introduce himself. Joe.
Thanks, Liz. Happy to be here. Excited for the presentations today. My name is Joe Tyler. I was diagnosed with FA, Friedreich's ataxia, about 8 years ago at WashU in St. Louis. That's where my dreams of being the first dual athlete, the dual athlete on the PGA Tour and the WWE, so a pro wrestler and a pro golfer, were diminished when I was diagnosed with FA. Moved on to bigger and better things. So it taking me all the way here. Kyle Bryant asked me to be here today. I lost a bet to Kyle. He told me I had to wear this hat throughout the entire presentation. I'm going to try to do that and try to keep things on track, try to, moderate this to the best of my abilities. So I'm extremely excited. Thanks, Liz.
Thank you, Joe. Thank you for sharing that. And I'm glad you lost the bet. Okay, so let's get started. Our first presenter today is Frances Smith. She's a grad student with Daniel Kozman at the University of Buffalo. And she's going to talk about molecular defects in Friedreich's ataxia, pathophysiological cytoskeletal abnormalities.
All right. Thank you, Liz. Can I get a thumbs up if you can see my screen, please? Awesome, thank you. All right, so yes, thank you everybody for joining me today. My name is Frances Smith and I work under the mentorship of Dr. Dan Kozman, and we're in the biochemistry department at the University of Buffalo. You might not have heard of us before specific to the world of Friedreich's ataxia, and that's because we're actually a lab that classically studies the presentation of iron in the brain. And so now it makes a little more sense of why I'm here, based on the presence of the iron accumulation in the cerebellar dentate nuclei of Friedreich's ataxia patients. Usually this is something that happens early on in disease, and it's often also used as a biomarker, so it's quite important. But what the literature lacks in Friedreich's ataxia is the cause of this brain iron accumulation and where it could be coming from. Now, specifically in the Kozman lab, we focus on a brain tissue system called the blood-brain barrier, and that's exactly what it sounds like. It's the network of vasculature that runs throughout the brain, and it has 2 very important jobs. First of all, it provides nutrition to the deeper parts of the brain and also prevents the allowance of toxins from the blood into the brain, which is a sterile environment. But what does that look like on a molecular level? Well, the healthy cells of the blood-brain barrier have proteins in between neighboring cells that act as a molecular zipper, and they make sure that the cells are zipped up tight and that they are a strong barrier so that nothing can move in between them. But we know in the literature of the blood-brain barrier that when those cells start to get sick, the zipper could start to come down and we could get some permeability or some movement of solutes from the brain, solutes from the blood into the brain when those cells start to unzip. But what causes this unzipping? We know that a change in the metabolism or an inhibition of the mitochondrial metabolism will do this, and so will the presence of dangerous oxidants signaling throughout the cell. But why am I telling you this at a Friedreich's ataxia webinar? Well, we know that these are two things that also happen in Friedreich's ataxia. Friedreich's ataxia cells have low energy metabolism, but there's also an excess of reactive oxygen species, which causes that dangerous oxidant signaling throughout the cell. And furthermore, our body's natural response to oxidants, which are antioxidants, is inhibited in Friedreich's ataxia. Now, just like how humans have a skeleton, cells also have a skeleton, which helps keep them functioning and structurally strong. And this is called the cytoskeleton, cyto being Greek for cell. Now, in non-Friedreich's ataxia patients, the cytoskeleton has tiny components like bones that fit together, and these are called actin proteins. But we know from some literature in Friedreich's ataxia that the actin proteins are actually structurally changed in disease, and so they don't fit together like strong bones anymore. And this does actually change the structure of the cells and the integrity of the cytoskeleton. So could this have any implications on blood-brain barrier unzipping? I actually have some very new and very exciting preliminary data that might show this case. On the y-axis of my graph here, we're looking at the strength of the blood-brain barrier model system. And we see that in cells that have normal frataxin here in the first column, they form a very strong barrier. But when we start to lose some frataxin levels and we make this Friedreich's ataxia model, we actually do significantly lose some of that barrier strength. But these are just cells in a dish. What will happen if we actually look at a real whole body system of Friedreich's ataxia? Well, that's what's coming up next. I'm actually very excited to soon get in a mouse model of Friedreich's ataxia and first of all, monitor their brain iron accumulation and also monitor the blood-brain barrier permeability using an MRI machine, just like how patients are diagnosed. So I want to tell you all that this is my premise, that unzipping of the blood-brain barrier could cause that early brain iron accumulation in FA. And if we start to understand this, could it help us learn more about the early stages of disease and possibly work on a new therapeutic window? I want you all to know that I'm very passionate about understanding this premise, as it's what I'm earning my doctoral degree in, and it's what the focus of my thesis is. So with this, I would like to thank FARA for the wonderful opportunity, but more so I'd like to thank the FARA Flash Talk audience, for without you, this opportunity would not be possible. So thank you very much everyone for listening, and I'm now happy to take questions.
Thank you, Frances. That was awesome. So the first question from the chat box: why does iron preferentially accumulate in, I'm going to butcher this, dentate nucleus?
That is an excellent question, and it is another unknown in the Friedreich's ataxia literature. A lot of researchers have speculated that this structure is very high in iron to begin with, so maybe just getting a little bit more in causes a lot of problems. And usually that's what we consider to cause the neurodegeneration that leads to the ataxic phenotype. Unfortunately, at this time, it is unknown, but if I recall, some other neurodegenerative disorders also have preferential accumulation in that area as well. So it's a great thing. I hope we eventually figure it out.
Frances, I received my degree in economics, so I'm going to ask a lay question here.
Please.
I had to have 14 cups of coffee to be this energized today. I look like that picture. Why, very simple, why couldn't we take more antioxidants to assist with that?
That's a great question. That seems, it seems like it should work, right? We actually have tried that in some clinical trials, and unfortunately, any of the clinical trials that we have in FA at the time just haven't reached, you know, a productive endpoint. I wish I had an answer for you. That, you know, the interplay of metabolism in FA is so diverse and so vast. Gosh, I have no idea, but I wish it worked. It seems like the best kind of target out there, right? But.
And we didn't, at least I didn't think you would come to the talk today with a cure, but if, you know, if you could.
God, I wish.
Yeah. Liz, any questions?
I was just curious. So the idea is that you have accumulation of iron in the cell because you don't have the, you know, functioning iron or formation of iron cluster. And then on top of that, you accumulate the iron. That's your hypothesis because, you know, the blood-brain barrier is impaired. So what do you think happens in the heart there? You know, why do you have accumulation of iron? It's just, you know, just the cellular accumulation of iron in the heart that causes the disease?
That's a great question. We've actually never worked on a heart model before, but it is very interesting. And obviously with the cardiomyopathy, that's something that's very important to look at. I've always found it interesting because based on my hypothesis of a loss of the blood-brain barrier integrity versus the fibrosis of the heart, so the thickening of the heart wall, it's almost opposite, right? So I've always wondered, what could the changes be if there is a change in the cytoskeleton that's causing those differences? You know, why would it be different in one tissue system versus the other? I do tend to side with the heart researchers who think that it is a cellular iron accumulation in the heart, just based off of how metabolically active they are. The brain endothelial cells that we have actually don't rely on OXPHOS, on mitochondrial metabolism that much. They really they work mostly off of glycolysis, but they are about 40% dependent on mitometabolism. So it will certainly be interesting and, you know, important to compare between the brain and the heart. Great.
Thank you, Frances.
Welcome.
Okay, let's move on to the second speaker, Anna Maria Schreiber. She is a grad student at the University of Alabama at Birmingham with Merrick and Jill Napierala. And she's going to talk today about Friedreich's ataxia neuronal progenitor cell model containing an endogenous frataxin luciferase fusion gene as a novel screening platform. Anna?
Can everybody see my slide?
Yes.
Okay, awesome. Thank you so much for the introduction. I'm Anna. I'm a PhD student at Marek and Jill Napierala's lab at UAB. And today I would like to talk to you about how can we actually use Friedreich's ataxia patient cells for the potential drug screening. So sometimes patients are kind enough to donate some biopsies for us. And from biopsy, we are able to get the fibroblast cells. So fibroblast cells are the cells that are actually making your skin, and they already have a defined function. But we have a special system that we can basically reprogram those fibroblast cells to something called induced pluripotent stem cells. And induced pluripotent stem cells are really interesting because those cells don't have a defined function yet. So they're basically like a blank canvas for us. And they also have a special function that we can differentiate or change them into different types of cells and give them a function. For example, we can change induced pluripotent stem cells into different types of neurons, heart cells, or pancreas cells. So now let's talk about how can we efficiently screen for the potential drugs that can contribute to the treatment of Friedreich's ataxia. So generally, talking about drug discovery, it can be a time-consuming and laborious and intensive process. So we need to conduct a lot of experiments in a span of time, and sometimes they are just not efficient enough. But we can take a different approach and create something called a reporter cell line. So reporter cell line is a special cell line that has one distinctive feature, and it's called a reporter gene. So whenever we are using a reporter gene, we can monitor its expression by adding a special substrate, and the expression is registered as a light peak. So whenever we put a reporter gene within the frataxin gene, we can actually monitor for frataxin expression, how the levels of frataxin are changing just by registering a light peak on a special machine. So the reporter gene reports for the frataxin expression. So we decided to create a novel reporter cell line using the induced pluripotent stem cells by putting the reporter gene using a CRISPR-Cas9 system, which is a system that acts like a molecular scissors. So because we are using induced pluripotent stem cells in our reporter cell line, we are able to change them or differentiate them into different type of cells, as I mentioned before. So we can take our induced pluripotent stem cells and we actually change them into cells called neural progenitor cells. And NPCs are really important cells because they are giving the rise to the cells of the central nervous system. And what's most important to this reporter cell line system, whenever we change the induced pluripotent stem cells into the NPCs, they still have the reporter gene within the frataxin gene. So now let's imagine a situation how we can utilize our reporter cell line for looking for potential drug candidates. So we can perform different treatments using our frataxin reporter. So let's imagine two situations, whenever we have a control and whenever we have a different type of compounds that we want to use for the potential treatment. So in the example one, we can see that the control and drug doesn't differ really much. So whenever we're using our reporter cell line, the light peaks between those two samples wouldn't differ. But whenever we have a compound that's able to stimulate the frataxin gene expression, as we can see in the example 2, we can see there is a high difference between control and the frataxin expression because machine is registering a high peak of light within the compound that's able to stimulate the frataxin expression. So because we can use the reporter cell line, we are able to screen for the 1,000 compounds in just one single experiment. And this concept that I'm showing you here, we actually tested. So as a proof of concept, we actually screened a library of 288 compounds, and we were able to prove that our reporter cell line is sensitive enough and can differentiate between the compounds that are not stimulating the frataxin expression and the one that give rise to the expression of frataxin. I'm really excited that I had the opportunity to talk to you today, and I hope we can utilize our reporter cell line for the future research that we are working on right now, and I'm happy to answer your questions right now.
Anna, that was cool. Coming from a patient's perspective, so biopsies hurt, right? They're not fun to give. I gave one quite a while ago. Would you encourage all patients when they're asked to provide a biopsy, if they can?
First of all, thank you for all the donations from all the patients. It is really important from a perspective of a researcher to get those biopsies because Friedreich's ataxia patients are highly heterogeneous. So you all have the expanded GA repeats, but they will differ in length on both of the alleles, and they are really important material to study. Because Friedreich's ataxia is also a disease, as you know, that will manifest in certain type of cells, like your neurons or your cells or the cells of the heart. And we are not able to really study those cells as they are. And just having the biopsy and having the possibility of turning those cells into induced pluripotent stem cells and further into the different cell type, for example, neurons or the cells of the heart, we are able to actually study the cells that are relevant to the disorder. So yes, I would say if you can donate, we are really appreciating it and you are contributing to all of our research.
Yeah, that I think all of us need to participate there. I definitely agree. Sanjay had a question. First, nice talk. Where is the reporter inserted relative to the GAA repeat? And is it on both genes?
Thank you for the question. So our knock-in of the reporter is actually only on one allele, and the reporter is inserted right behind the 5th exon of the frataxin.
So I have a question. So do you know if that frataxin is functional? The one that expresses, that is fused?
The pipeline of my experiments, of course, whenever we are just using the reporter cell line, our knock-in is an luciferase. So whenever I'm performing a luciferase assay, I'm only able to read out the luciferase expression. But whenever I was validating the compounds that we used for our initial screen, I can see the results being mimicked on the levels of the transcript and on the levels of the protein. So I would say it's pretty accurate in terms of predicting the stimulators.
I think we have another question, right?
This next question, Anna, comes from Ron Bartek, who's here today. Thank you, Ron. Has your screening of compounds already identified compounds that might be effective in elevating frataxin protein levels? If so, what is the nature of those compounds?
Thank you for the question. Yes, from my initial screen and for the initial experiments that I performed, we were able to identify a few of them that were actually increasing the frataxin levels on the protein. And the nature of the compounds is, broadly talking, epigenetics, and they are the histone deacetylase inhibitors, but the types of that haven't been previously tested in Friedreich's ataxia context.
So did you find compounds that increase the mRNA by a lot, but not the luciferase signal? The mRNA, but not the protein?
I don't think that I had a situation of just mRNA, not the protein. I had a situation of there were hits on my initial luciferase screen, but when I moved my research into the transcript and into the protein, it turns out they didn't have that much of an effect. But I personally cannot recollect an experiment that it was just mRNA. So it was, whenever I was looking, it was both mRNA and protein.
Great. Thank you, Anna.
Thank you so much.
Okay, the next speaker is Eva Zubanovic, and she's a research fellow at the University of Melbourne with Cristina Cortez-Hugo. And she's going to talk about sugar-based nanoparticles for frataxin delivery.
Hello, can you see my screen?
Yeah.
Okay. Thank you for the opportunity to talk to you today. I'd like to present our approach for gene therapies for Friedreich ataxia. Gene therapies are of interest in nanomedicine as they allow potential treatment of diseases that are not treatable with currently available methods. And for Friedreich ataxia, this could include delivery of frataxin encoding gene, typically in form of DNA. The challenge here lies in the nature of DNA. It is a molecule prone to degradation by enzymes present in our body, and it's also negatively charged, which prevents it from freely crossing most of the biological membranes, which are also negatively charged. So successful gene delivery requires formulation of gene carriers. And there's been considerable effort towards development of viral-based carriers. We are working on alternative, which is development of nanoparticles that can encapsulate frataxin gene. There is a wide choice of materials available to prepare nanoparticles, which allows us to tailor it for Friedreich ataxia application. And we can also modify particle surface so it binds specifically to the population of cells of interest. We're currently working with a biopolymer called glycogen, which is a natural polysaccharide, in other words, sugar, that is acting as energy storage in animals and plants. It is an extremely convenient material to work with because it naturally exists as a nanoparticle. It is around 30 to 80 nanometers in size. So we chemically modify it so it has a positively charged binding moieties to complex frataxin DNA. So just by simple incubation together, we obtain soft self-assembled nanoparticles, which are around 180 nanometers in size. After characterization of such system, we're extremely interested to test our carrier in a clinically relevant model. And this is possible thanks to recent advances in stem cell technologies. We are working with stem cells obtained from fibroblasts from patients that were differentiated into neurons. This gives us unique opportunity to work with Friedreich ataxia neuronal model that not only carries a genetic profile of the disease, but that can also be cultured in 3D. Therefore, it's a model that mimics the complexity of interconnecting neurons in human nervous system. Additionally, our model cargo is gene encoding for frataxin, but also gene green fluorescent protein, which allows us to track our progress and screen the drug delivery carriers using fluorescent microscopy. So I selected this picture because it happens confocal microscopy is one of my favorite techniques. But I also want to highlight that we're not only able to say and quantify by different methods what's the percentage of cells that were successfully transfected with this gene, but we're also able to assess how far the particles were able to penetrate such 3D structures. And it's extremely valuable for us because this allows us to identify the properties of carrier that have a chance to be effective in delivering frataxin gene. So what I'd like to highlight is one that we are developing nanoparticle-based gene delivery systems for Friedreich ataxia, but also I'd like to highlight that this work demonstrates the combined effort of material chemists and stem cell biologists and presents the patient-derived cell models as platform for screening various drug formulation for Friedreich ataxia, including nanoparticle-based carriers. Thank you for the opportunity to talk. I'd like to acknowledge all of our team members and FARA. Thank you, and I'll welcome all the questions.
Thank you. And is it pronounced Eva? Or?
Yes, that's correct.
So glycogen, that's essentially carbohydrates, right?
Yes.
Okay. Could the body potentially reject that as like an invasive, I guess, is that the right term?
Yeah, thank you for that. Actually, there's always a concern about researchers in our field that the particles could be recognized as a foreign body, as you suggest, and be cleared. But the cool thing about glycogen is that it's a natural origin, and it is present naturally, so there's less chance of rejecting it. And also, we can chemically engineer it, adding PEG, for example, molecule on the surface that can kind of help it to not be visible for the immune system as a foreign particle. So there's definitely need for further research into that, but I think it's possible to mask it as something friendly. And
Yes.
our initial studies also show there is no cytotoxicity when we test it in vitro. So,
Paul brought up a great question. What are the advantages of your approach versus the Larimar Therapeutics TAT approach to deliver frataxin? Or Liz, did you want to address that?
Yeah, so the TAT frataxin, the therapeutic approach that is used to deliver frataxin directly to the cell. So maybe there are two parts of the question, this question. One, if your approach can also be used to deliver protein instead of nucleic acid. And two, maybe what are the advantages of the nanoparticle versus other ways of delivering nucleic acid, for example, with viruses?
Thank you. So regarding the first question, or the first part, potentially when we're working at the DNA level, we are looking at integration of the DNA into the cell. So we're talking here potentially about long-term and sustained expression of frataxin that would not require reapplication of whatever drug we choose, which may not be the case for protein. And there are also very different in terms of delivering it, different materials, the proteins and the genes. And that kind of leads to the second part of the question. I think it's just an alternative. It's a different way. None of them is worse or better. It's just we're trying to look at this from different angles. And I hope together we can find out a solution.
Jay has a question. What happens to intracellular nanoparticles over time? Oh, Liz wants to answer. Sorry, Liz.
No, I didn't say it.
So actually, we do a lot of research into that, into understanding as well how our particles process inside the cell. And our study shows that this is the particle is mainly over time degraded, but the goal it has to meet, so the delivery of the gene is met. So in other words, what we're relying on is the particle to protect the frataxin gene, deliver it into the cell and release it, where it can then be processed by the cellular machinery. But the material itself is slowly degraded, but it's just a natural process. And that's actually a positive outcome for us. We track that mainly using super resolution microscope and trying to really understand what's happening.
Liz, Alex's question?
Yeah, sure. Go ahead. We have a minute.
How effective are nanoparticles at delivering genetic material versus viral vectors? And that's from Alex. He says, thank you, and thanks for the great work.
I can only speak at the pre-discovery stage where we are, where we are at. So we are achieving, for example, this, which I had on slide, which is our currently lead candidate. It's a corn-derived glycogen, so plant-derived. 6 days post-treatment, we have around 50% of cells that express frataxin. And yes, this is something we need to look at long-term. We're also interested to kind of find a middle ground. We don't want the frataxin to go too high, as it could be toxic in excess, but So it's a work in progress. I'm sorry I can't directly compare, but the reality is we are at different stages compared to viral vectors, but I hope we'll get there.
Thank you, Eva.
Thank you.
Okay, we'll move on to the next speaker, Ana Luisa de Carvalho Cardozo Hernandez. She's from the University of Campinas, working with Marcondes França, and she's going to talk about spinal cord diffusion tensor imaging in Friedreich's ataxia.
Hello, my name is Anna. I'm a first-year neurology resident. I'm a former medical student at the State University of Campinas, where I did this work with my team. So firstly, I would like to say, for long now, we know about some regions of our nervous systems that are affected by Friedreich's ataxia. One of the main ones we know of is the spinal cord. Previous studies with autopsies have shown damage in a few specific regions in the spinal cord. I like to highlight two, which are the main focus of my work. The first one is the back part of our spinal cord, which is called the dorsal columns, which is where we have the sensations such as proprioception, which is the sensation we have to know where each part of our body really is, that helps us coordinate movement. And it's one of the causes when we lose this ability, then we develop ataxia. We also have damage in this region that we call the lateral cortical spinal tract, that is responsible for the control of movements of our limbs. And we know that both these regions and other regions as well are severely affected by Friedreich's ataxia. But we know this from previous autopsy studies. The new tendencies for us is to find markers while in vivo to find those differences and those damaging partners in patients with Friedreich's ataxia. Now, we have previous studies with MRI that have showed that patients with Friedreich's ataxia have a spinal cord thinning, but they have not studied the specific regions inside the spinal cords. And also the methods they use are not automatic. They are semi-automatic methods or manual methods. So they have a big inter-examiner, they are not reliable between different people who are examining this imaging. So we propose a study using this new tool. It's called the Spinal Cord Toolbox. It uses the spinal cord MRI, and we can segment different parts of the spinal cord using DTI, which is a method that measures the diffusion of water molecules throughout neuron fibers to assess damage in the white matter. And also it also measures the whole spinal cord area. We can see here this method. They also segment the spinal cord levels automatically. It's a very not time-consuming protocol. It's almost 30 minutes or so, and patients normally tolerate it well. So we did we compared patients with controls to see the specific damage in those regions I just mentioned. So the lateral cortical spinal tract, the dorsal columns, whole corticospinal area, and the whole white matter tracts. Okay. What we found was that the difference between patients and controls was significant with this method. So we could see differences, but we also wanted to investigate more. So we wanted to see which marker could be the best one for us to assess difference between patients and controls. This is important because this can help us use this as a tool to diagnose patients. So if we know that patients have this marker as a sensible marker for this disease, this can help us differentiate from other forms of ataxia. So we measure the effect size between the differences in patients and controls. And we found that the damage in the dorsal columns in a specific was the damage that we found the biggest effect size, so the biggest difference, which means this could be a parameter we can help diagnose patients with and differentiate other forms of ataxia. We also investigated the correlation between the imaging findings we found with parameters of the disease, which means disease progression, disease severity. We also looked at age of onset, and we found that most of those parameters correlated with the imaging parameters we found, mostly with the damage we found in this back part of the spinal cord, the dorsal columns, and also, with the lateral cortical spinal tract, I'm sorry. and also with the whole spinal cord area. We still don't know which one would be the most reliable because both of them correlate well, mainly with disease severity. But that is important because when we do a clinical trial to investigate treatment, we also want to look at disease progression to see if this treatment is indeed slowing the progression of the disease. And this marker seems as good candidates to show us that can be used by clinicians in the future to evaluate the progression of the disease as an extra data. So I think the main goal of our study was accomplished in the way that this is a more initial study to search for candidates. And I think we found a good candidate for a diagnostic tool for evaluation of disease progression. And with using a tool that is a reliable tool, automatic tool, that is also not time consuming and it's actually pretty easy for patients to tolerate. So I want to thank you all. I've been doing research now in Friedreich ataxia for a couple of years, and I really enjoy to be a part of all efforts in the community to help find hope for our patients. And I'm disposed for any doubts that may present. Thank you.
Anna, thank you. Any other biomarker that we can have other than the 9-hole peg test patients are happy about? My only question, so I've done studies at the University of Minnesota, the MRI studies. My question is around the MRI machine itself. Do you need a stronger MRI machine to see these changes, or any hospital MRI machine will do?
So we have different types of MRI machines. We use a 3 Tesla MRI. There's 1.5, but a lot of hospitals, they already use the 3 Tesla. There's higher precision machines as well. But this actually, a lot of the machines can do this protocol, and it's very easy to adapt to the system.
Cool. Oops. Sanjay has a question. How early do these cervical cord changes start? What was the youngest patient's age? And could some of these changes be developmental?
I'm sorry, the last one. Can you repeat the last part?
Could some of these changes be developmental?
Okay, so that is a big question. The second question is a big question that we are asking ourselves. So I can't tell you exactly which one is exactly the one that the changes are fine, are the youngest patients, because we did a patient control analysis. So we didn't determine a range which below this we would consider it altered or not. So I cannot tell you this, but I can tell you that I've seen all images, and it's pretty, you can see this from the image I showed. This is a patient and this is the control. It's pretty visual, the thinning of the spinal cord, and we can see, and I could see it in all images. And we have patients that are younger age. Our study, unfortunately, we did not have a significant part of younger patients. That was, I think, a restriction of our results, but I did see changes in all patients. The developmental changes, this is a topic. Our group has already made previous studies regarding this. What we think may be some of the findings of this study have shown this is we found a difference between the damage in upper cord levels like C2 and C3 and C4 and C5. C4 and C5, there were bigger damage. We could see that there were lower values, and we compared between levels. We could also see that C2 and C3, they were more correlated. They were the best levels to correlate with the disease parameters, which may show that the damage in those upper cervical levels are more related to the course of the disease. But it's still very early to reassure. I think we would need a longitudinal study to really see if those changes are happening over time.
So Anna, why did you choose the cervical cord, and can maybe differences in the lower part of the spinal cord be bigger or better?
Yes, we do know that from previous pathological studies from the autopsies, we know that the transition from cervical to thoracic spinal cord is very affected in the disease. So the lower damage also is compatible with that. The problem with the thoracic region is that sometimes it's harder for imaging methods to see the differences, because the biggest challenge for spinal cord MRI is because patients can move during the exam, and lower parts of their spinal cord are more sensible to those movements. So if we could find markers in the cervical regions, I think is something that it's easier to be used in a broader spectrum.
And I think you mentioned a correlation with, disease duration or disease onset. What about GAA repeats?
We have not studied this. We did not have the data from all patients, because a lot of those patients had already done the tests outside of our hospital. So we had from a limited number of patients. This is also some type of analysis we are planning on doing on future studies as we continue to gather data.
Great. Thank you, Anna. And we are at the last speaker for the day, Julia Dunn. She's at Children's Hospital of Philadelphia and works with Shannon McCormack. And she's going to talk about bone health in Friedreich's ataxia.
Hello, let me share my screen. Can everyone see that all right?
Yep.
Cool. Thank you for the intro. Hi, my name is Julia Dunn. I'm a rising senior studying bioengineering at the University of Pennsylvania, and I've been doing research on Friedreich's ataxia for the past 18 months. I started by doing some imaging work, and lately I've been looking at bone health in FA, which is what I'm going to talk to you guys about today. We wanted to study this topic because clinically our team has encountered individuals with FA with osteoporosis and fractures, but it hasn't been studied broadly. We also think these complications are likely to be more widespread because in other neuromuscular disorders, impaired bone health, which can increase your risk of fracturing and osteoporosis, is a common complication. So we wanted to look at this subject specifically in FA. As some background, we quantify bone health using bone mineral density, so how much mineral content is in your bones, and a clinical tool called a dual energy X-ray absorptiometry scan, abbreviated as DEXA, measures this BMD. For children and adults under 55, BMD is reported as a Z-score, and this tells you how many standard deviations above or below the mean your BMD is compared to people your same age, race, and sex. So for a person to have a Z-score less than -2, which means more than 2 standard deviations below the mean, that means that they have low BMD. So to try to learn more about the burden of impaired bone health in FA, we looked to 2 different sources of data. First, we looked at 3 different studies done at CHOP where DEXA scans were an outcome of the study. So this included 19 healthy control participants who were of similar age, sex, and weight to 19 adult individuals with FA. And there were also 7 children with FA in these studies. The second source of data was from a survey on bone health and diabetes in FA, which was designed by Dr. Jacqueline Tamaroff, one of my mentors. And she actually introduced this survey at these Flash Talks last year. So a huge thank you to you guys for filling it out and helping us generate this data set. She sent it out to over 400 people with FA at CHOP and received 133 responses, 105 of whom were adults and 28 of whom were children. So down here on the left in panel B, this box plot shows the bone mineral density of the 7 children from the retrospective studies. If these children had the same BMD as their reference population, the average Z-score would be centered around 0. However, we can see that that is not the case. The Z-scores for both the whole body and the femoral neck, which is in your hip, are pretty low, and the average whole body Z-score is less than -2, which we said earlier was the cutoff for low BMD. We also see a similar pattern in adults from the same studies. So as you can see for the individuals with FA, the average is once again lower than you would expect to see if they had the same BMD as their reference population. Not only that, but we found statistically significant differences between the average Z-scores of the 19 individuals with FA and the 19 matched healthy controls from the study. So from these box plots, we can see that individuals with FA have lower BMD than healthy reference populations. And we wanted to look at clinical factors that could be related to bone density. And from that, in panel C, we found a statistically significant correlation of negative 0.64 between a patient's femoral neck Z-score and modified FARS score, or mFARS, which is a clinical marker of disease severity. We can't say anything about the directionality of this correlation, but a possible explanation of the result is that severe FA is causing both the higher mFARS scores and the lower BMD. People with more severe FA are likely engaging in less weight-bearing physical activity, which can lead to lower BMD. And then last but not least, panel D describes some of the data from the survey on fracturing. Of the 133 respondents, 67 reported using a wheelchair as their primary ambulatory device. So the data you see here is only from those 67 people, as they are at greater risk for low bone mineral density. 34 of the 67 people reporting wheelchair use also reported having had at least one fracture in their lifetime. A breakdown of how they sustain these fractures is in the top pie chart up here. But not every type of fall or fracture is the same. So we excluded all reported fractures of fingers or toes because they're easy for anyone to break regardless of the state of your bone health. So it's hard to tell which of these fractures would actually pose concern. Also, the more low impact the cause of the fracture, the more concerning or potentially indicative of low bone health the fracture is. So from these options available on the survey, falls from standing height, falls from below standing height, and incidentally detected fractures are the types of fractures that would warrant the greatest concern. So these three categories shown in orange, gray, and yellow on the pie chart account for about 63% of all of the reported fractures. And the second pie chart below is representing what DEXA scans are showing in individuals who are using a wheelchair. And for the most part, we don't know what these DEXA scans are showing because only 11 out of the 67 respondents reported having had a DEXA. So what does all of this mean? We can see that individuals with FA are experiencing low BMD and that it is associated with disease severity, but the minority of people are undergoing DEXA scans. Information on when you might consider getting a DEXA, the potential roles of calcium and vitamin D to help bone health, and the potential use of anti-osteoporosis medications will be included in the newest 2021 FA guidelines, which is exciting, but there are clearly some missed opportunities to screen and find treatable low BMD in individuals with FA using DEXA scans. Thank you very much, and I can take any questions.
I don't know if Liz purposely put you in this position, in the last position on purpose, but Julia, my father-in-law accidentally ran me over with his car back in February, and I broke my first metacarpal from falling down from him running over my foot.
Wow.
I've broken many bones. And I often wonder, like, why am I more susceptible to broken bones than my friends? Am I clumsier? Is it my bone health? What is it? So this is super fascinating, super cool. Do you think, and again, this is going to be a very lay question, should we drink more milk? Should we? What can we do right now?
Yeah, so I want to start by saying that I'm not a clinician, but my mentors both are. And so we have talked about this. So some good options if you want to improve your bone health, weight bearing exercise, if you talk to your doctor first, is a good way to improve your bone health. You also should avoid smoking. And if you have diabetes, make sure to manage that and make sure that you have enough calcium and vitamin D in your diet.
Yeah. Cool.
So Julia, I was curious about iron. So is iron overload kind of a possible mechanism that causes some of the bone loss?
That is a really good question. One I'm not sure the answer to, but I could definitely think more about that and let you know, because that that's a really good idea and something that we haven't been considering when we've been looking at this data.
And also another question regarding calcium dyshomeostasis, you know, that's been shown in neurons, and that might correlate to some damage in that you have in the neural tissue. Do you think calcium dyshomeostasis is also part of, you know, what happens in the bone?
That's also a really good question, and one that we haven't been considering, but super interesting. And again, sorry, but we can definitely think about that more, and I can let you know.
Great. I don't see any other questions. So with that, I am going to thank everybody today for speaking and sharing your research and answering your questions. Oh, there was a question, I think. Oh, two.
I think they're just thank yous, Liz.
Oh, okay.
I do want to say to the panel from a patient's perspective, and I'll try not to Bartek this up and start crying, but thank you, because it means so much to us. So thanks.
Yeah, thank you.
I Bartek it up.
That's a new verb. I've never heard of it, but I'm going to use it from now on. So thanks everybody, and thank you, Joe, for moderating and for Barteking. And also, I'm a little disappointed that you didn't wear the hat all the way through, but that's okay. Thank you for all who connected today, for participating and for asking questions. Remember to fill out the survey and vote for your favorite flash doc. And don't miss our last webinar. It's going to be next Thursday at noon Eastern, and see you then. Thank you all.